Earlier I have already discussed that what is the structure, morphology or composition of biological cell. I also discussed that what is the properties of the cells like cellular adaptation like atrophy, hypertrophy, metaplasia, hyperplasia. I also discussed that that how protein, what is the structure of the protein molecules like amino acids, and how these protein molecules are formed and how protein molecules they interact with each other, and as well as the collagen, what is the composition of the ECM extracellular matrix and so on. And at the same time I have focused on the part, the tissue, that bone is an example of a hard tissue. What is the composition of the bone? This is a combination of the mineral phase, that is 69%. So 69% means that your natural hydroxyapatite or calcium phosphate particles which are in nano size in the bone, which is contain 69 weight%, a majority hydroxyapatite as I said. Then you have citrate, carbonate, fluoride and hydroxy ions. Then you have organic phase, and organic phase is 22 weight%, and water is your 9 weight%. Now among this organic phase your collagen is 90 to 96 weight%. That means majority of the organic phase or most of the organic phase is your collagen. So that is the reason bone is a composite, and this composite is known as collagen hydroxyapatite composite. So collagen is your organic phase, so that's like a polymer. Hydroxyapatite is ceramic phase. In other words, you can describe bone as an unique example of the polymer ceramic composite. Now hydroxyapatite crystals that form a slender needles in the collagen fiber matrix, and the resulting mineral containing fibrils form lamellar sheets. Now let us see that what are the different examples of the bones based on their shape or size. Now these typical bones, they consist of cortical bone, cancellous bone and bone marrow. So I think in one of the earlier lectures I have mentioned that cortical bone is the outer surface of the bone which is very hard and strong. Cancellous bone is the porous structure or called trabecular bone. So porous structure means whenever bone has a porosity, that means the properties are inferior compared to the cortical bone. So property wise cortical bone is stronger compared to the cancellous bone. Then you have the bone marrow. Bone marrow is that region or matrix region inside the bone. Now many times if you say that a patient is having a cancer and people say bone marrow cancer. Bone marrow cancer means there are some cells inside the bone marrow region that is proliferating at an abnormal pace. So cancer is nothing but cell growth or abnormal growth of a group of cells, and which cannot be controlled or which is not natural. Long bones. Long bones is typically in tubular in shape, and this length of the bone is greater than the breadth. So if you consider the aspect ratio of the long bone is much greater than 1. That is the length to the breadth ratio. Some long bones can actually be short, and this consists of a shaft and two enlarged curved ends, and shaft of the bone consists of cortical bone surrounding medullary cavity. I think I will show you some slide what is meant by medullary cavity. Now examples of the long bones are the thigh bone, that is called femur, tibia and fibula, that is the calf bone, humerus, that is the upper arm, and radius and ulna is the lower arm. So you have the femur, tibia and fibula, and you have the humerus, and you have the radius and ulna. Now let us see that how this macro structure of the long bone they look like. Now this is a typical structure of a humerus, and if you see that humerus entire structure, you have a kind of a cap here, and you have the long end also. So you have a cap, that is that long compact bone, and compact bone is the outer surface of the compact bone, and inner surface you have a spongy bone. Spongy bone means cancellous bone. So cancellous bone has a different name. One is the spongy bone because it has a porous structure. Other name of the cancellous bone is called trabecular bone. So trabecular bone, spongy bone, cancellous bone, they are synonymous. Synonymous means they carry same meaning. Now cortical bone, that is the harder part of the bone. Now if you take a cross section, what you can see in the cross section, this is called your actually medullary cavity. Now through this medullary cavity you have a blood, proteins, everything they are circulated throughout, because blood is the tissue which carry always oxygen to different parts. So if the blood flow is not there at any part, that means oxygen is not transported. If oxygen is not transported, cells will die, cells will undergo apoptosis, and therefore tissue will not perform its automatic function, tissue also will die. Now epiphysis is the end of a long bone, and this is the, you can see that is that how this long bone end will look like. So this is the region I am now, so this is the region we are now focusing here, and these are different parts. That A is the articular cartilage at the end of the bone. So this region is that articular cartilage. Now B is that bone trabecule or the spongy bone. Now C is the red marrow cavity, that is the region that is red marrow cavity, and D is the hyaline cartilage. Now D is again this part. So cartilage is another tissue. So what it shows here, both the outer region and the inner region you have the cartilage tissue, and inner part you have that cartilage cancellous bone or the trabecular bone. Then what is known as the flat bone? Flat bones is that calvaria, and irregular bones are the facial bones. So facial bones you can see that is in the skull, that is called the facial bones or irregular bones. These are examples of the irregular bones, and flat bones, the name is that called now short bones typically are cuboidal in shape. If you remember that how these bones are classified into different types, long bones, if I can summarize, long bones are actually length is much greater than the width, that means aspect ratio is much greater than 1, and you have seen that long bones typically consist of cortical bone, trabecular bone or spongy bone, third one is that bone marrow region or medulla region. Then you have called flat bones, and you have called irregular bones. Then you have called short bones, typically it is cuboidal in shape, and it consists of the cortical shell with inner trabecular core. So typically what you see, generically bone always contains cortical shell, that means that is the hard surface, and the hard surface actually protects the internal part of the bone. So that is the reason, you know, that this concept is very natural. That is the reason people always put coatings on the surface. Coatings means the coatings are typically harder than the substrate. So whenever you put coatings like titanium nitride coatings or titanium or some other examples, then titanium nitride has much higher hardness, much better wear resistance than the titanium. So that is the reason that why coatings are placed on a soft substrate. Similarly, all the bones, they have a cortical shell like outer surface and inner trabecular core, and exist only in wrist and foot. So these are like short bones. Wrist as well as foot, they are called short bones, and examples are the carpal and tarsal bones. So carpal is any of the 8 small bones of the wrist, and tarsal bones are any of the bones of the ankle and the heel in the human being. Then you have called flat bones. They are essentially consist of 2 plates of cortical bone with porous tissue in between. Again, if you come back to that, just the description that I have mentioned to you, that is the generic description, that you have always the cortical bone which is at the outer surface, and inner material is always the cancellous bone. So that means the hard surface which is protecting the spongy material or spongy surface, and this is generally curved rather than the flat, and calvaria is the top of the skull, scapula is the shoulder blade. So that is also the examples of the flat bones, and the ribs are also another examples of the flat bones. Irregular bone, as the name suggests, irregular bones means it has a various shapes, and it also, the composition also depends on bone, and it consists of a thin cortical cell surrounding trabecular core. Again the same principle, some hard surface protecting the weaker region, that is the trabecular bone. And the examples are the facial bones and vertebrae, that is the bones of the spine. That means that is at your spinal cord or spine. These are the examples of the irregular bones. Irregular means that does not have any specific shape, like long bone or flat bone. They do not have that kind of specific shape, like cuboidal shape, that is short bone. So irregular bones, like it is always, it does not have any specific geometrical shape, let's put it that way, so that you understand that why it is meant by irregular shape. Now this is a sketch of the irregular bone structure. As you can see that it is really of different very irregular shape, and this shaped materials is called irregular shape. Now this slide actually gives you a table which gives you the range of properties that is typically measured with different parts of the bone, like cortical bone, cancellous bone, enamel and dentin. Enamel and dentin are the hard tissues of the teeth, right? Enamel is the outer part, dentin is the inner part. Similarly, cortical bone and cancellous bone, again is the cortical bone is the surface, cancellous bone is the inner core. Now, first thing I must mention here that you see all the ranges of values. Now, that means all these bone properties, they cannot be characterized by unique value, unique property value. You cannot say elastic modulus of the cortical bone is 10 gigapascal, because depending on the anatomical location, like whether you are taking from the wrist, whether you are taking from thigh, whether you are taking from leg, your bone property is also different, because the composition of the bone is also different depending on the anatomical location. And if the composition is different, composition means that is a proportion of the collagen and the synthetic and the collagen and the natural hydroxyapatite. If this composition varies, the composite properties depends on the ratio of the two phase, like alpha beta. If you change the alpha beta ratio, then the composite made of alpha and beta must change. Similar rule can also be applied to the bone, because bone is a polymer ceramic composite. So, if the ratio of the collagen to hydroxyapatite changes, that will also change the bone properties. Okay? And that is what I am trying to hint here, that this bone has a range of the properties, be it cortical bone, be it natural bone, be it enamel or dentine, and this range of properties is already shown here. Now, density of cortical bone is somewhere around 1.8 to 1.2, cancellous bone, because its porous in nature, is less, and density of enamel and dentine is 1.9 or 2.2. Now, when you talk about the elastic modulus, cortical bone has a 4 to 27 gigapascal, cancellous bone is much lower, because cancellous bone has a porosity, so the porosity reduces the elastic modulus. You have a compressive strength 10 to 160, cancellous bone can have a lower compressive strength. Then you have enamel has a 13.8 and dentine has a 20 to 84 gigapascal, which is close to 100 gigapascal. Okay. Now, as I was describing just now, that properties of tissue, some comments here that has been mentioned, that properties of tissues vary between individuals and with anatomic locations. That is what I just mentioned. And between individuals means like two persons of the same age can have different bone properties. Okay, that is what I am trying to say. Material properties are significantly affected by the degree of mineralization of the bone. Degree of mineralization means what is the mineral content, means what is the content of the hydroxyapatite in the collagen matrix. So, that is what is meant by degree of mineralization. The density of bone is not an intrinsic property and can change with time. For an old person, many times the doctor say the bone density is decreasing. Bone density is decreasing means number of bones at the unit length or along a particular length, that number of bones is reduced. Why? Because bones will slowly dissolve in the human body, so that total volume occupied by the bone is reduced, and that is called the bone loss. And that is why you can see that a aged person or a person of 70 years age, he cannot walk like a 10 years old or 20 years old person, right? Because that walking capability or running capability is reduced, because your tissues and the bones cannot perform the same function at the same speed like what a young person can do, because their tissue or the bone is losing its capability because of the bone loss or the bone density. Now, some more comments. Mineral stores within a bone can change dependent on the physiological demand. Mineral stores means again it is a hydroxyapatite content. So, it is essentially have. Now, pathological processes can affect the degree of mineralization. In normal and healthy bone, the range of bone and bone mineralization is small. What it means? In a normal and healthy person, the bone mineralization, that means synthetic or the amount of the hydroxyapatite content varies only within a small window. Let us say total hydroxyapatite content 69 weight percent. So, it can vary over 68 to 70 weight percent, not like 60 to 70 weight percent, that is a large window. Now, what is pathology? Pathos means like it derives from Greek words, like most of the things are derived from Greek words. Pathos means disease and logos means word reason. So, it is the study of the links between the disease and the basic sciences. And what is a disease? A disease is a physical or functional disorder of a normal body systems that places an individual at an increased risk of adverse consequences. So, like when you have fever, you cannot do your, or you cannot perform your normal functions at the same speed like when you are in healthy conditions, okay, because your tissues will not perform its normal function. So, because that there must be some functional or physical disorder of the normal body system, and diseases are typically diagnosed by physicians or other healthcare providers through a combination of tools. Now, whatever you have disease, that you can only get to know only when you do the blood test or urine test or other pathological test, okay. Now, let us discuss something on the mechanical response of the human cortical bone. Now, what you do, what has been plotted here, stress versus strain. Now, this is a very standard way of plotting the mechanical response of any material, let it be metal, ceramic, polymer, etc. So, that mechanical response is always expressed in the plot of stress versus strain, and that is what has been mentioned here. Now, this is a human compact bone, means compact means it is like a cortical bone, mostly it is a cortical bone. Now, what you can see from this graph you can see, if you increase the strain rate means if you increase the epsilon dot means this is d epsilon by dt, and that you can increase by varying the cross-head speed. Suppose you have a tensile specimen, you are pulling it with 2 cross-head speed, and that cross-head speed if you increase or decrease, or if you vary, then your strain rate also will vary. And if you increase the strain rate from 0.001 per second, here it is a flat one, to 1500 per second, what you see? You see that curves become much more steeper. Now, how this elastic modulus is calculated? Elastic modulus is calculated from the slope of the linear part, right? And from there what you can see, when the strain rate is increased, so if your epsilon increases, then your elastic modulus increases, right? When epsilon increases, your strength also, that is sigma t or whatever, that is also increases. But what decreases? That is your ductility, that is decreases. And how this ductility is measured here? Ductility is measured by the total strain at failure. Now, total strain at failure is roughly 1%, and when you are doing the test at 0.001, then your total strain is 1.6%. So, as you can see that it has a linear region followed by a flat region at the low strain rate. Similar thing is observed up to the strain rate of 1 per second, but when you go to very high strain rate experiments, then it becomes flat. So, then it becomes very steep, and the non-linear part or the flat part is also reduced, and accordingly your elastic modulus increases, your strength increases at the expense of decrease in the ductility. That means your strain to failure is significantly reduced compared to the low strain rate test. So, in other words, major message from this plot is that with increasing strain rate, your ductility decreases, and then mechanical properties of the mechanical response of the human compact bone is highly sensitive to strain rate. Okay. Again, mechanical properties of the human compact bone cannot be described by a unique value, because that depends on the strain rate, as you can see from this particular slide. Now, the question is that why it depends on the strain rate. The answer is, bone has a complex composition, like it has some water molecule about 9%. You have the majority is the hydroxyapatite, that is 69%. Then you have the collagen content around 22% or so. Now, it is a polymer ceramic composite. Now, polymers, the mechanical response of polymers are time dependent, okay, and that leads to a time dependent response of the bone, because bone essentially contains the polymeric molecules also. Because for ceramics, the mechanical response is not time dependent. You do the test at 0.001 per second, or you do the test at 1500 per second, I do not think the elastic modulus will vary much. But for polymers, it will vary, because polymer essentially the chain micromolecular kind of structure. The faster you do that, you are not giving enough time the chain reorientation to take place during the polymeric mechanical behavior, and that explains why bone has a time dependent mechanical response or strain rate dependent mechanical response. So, this list tells you actually some mechanical properties of the cardiovascular tissue. Now, what you can see here, this is the elastic modulus. Now, elastic modulus you have seen from metals and ceramic that elastic modulus is the level of gigapascal, 10 to the power 9 Pascal or 10 to the power 9 Newton per meter square. Now, what you see here, aorta for the cow or dog or thoracic aorta for dog or human, it is always around less than 2 megapascal. So, that means it is extremely soft tissue. Soft tissue means the properties are very much less. So, megapascal versus gigapascal means it is 3 orders of magnitude lower elastic modulus when you are considering the soft tissue. Now, this is the kind of some other complete range of the soft tissues that are mentioned here, like arterial wall. It has a maximum strength of around less than 2 megapascal. Maximum strain is 40 to 50%. Elastic modulus is 1 megapascal. It's so low. Tendons or ligaments, it has a maximum strength of around 200 megapascal, and this maximum strain is 5 to 50. So, what you can notice here that in both all these cases, at least these 3 cases, your maximum strain is less, greater or equal to 50%. So, it is a large strain of fracture, strain to fracture that is possible. You cannot get this kind of strain to failure in most of the metals or ceramic materials. Only in the polymers you can get this type of large strain to fracture. What it means? That means this most of the soft tissues are largely polymer based. That is why it gives such a large value of the strain to failure. I will be describing the typical protocol that is followed in vivo implantation as well as histopathological evaluation of biomaterials. So, before I do that, let me remind you that why in vivo testing is required, because in vivo testing is critically important, or it is very critical for development of clinical devices. For last few lectures I have described that in vitro test, like what is the in vitro biocompatibility assessment, or how different in vitro tests like cytotoxicity, genotoxicity, hemocompatibility, they can be performed in laboratory scale environments. And laboratory scale means that is the in vitro test. However, as I said before, in vitro test cannot replace in vivo test, because in the in vitro test you do not see any inflammation, and then second thing is that there is no inflammation and there is no immune response. So, because these tests, in vitro tests, are not carried out inside the animal model, so they cannot show, they cannot tell you, in vitro test cannot tell you whether a given biomaterial can cause inflammation when implanted inside the human body or any animal, or whether there will be any immune response to this biomaterial. Third important point is that these kind of in vitro cytotoxicity tests, they are always carried out with single cell type. Single cell type means like it may be either fibroblast cells, that is connective tissue cell lines, or it can be osteoblast cells, that is the bone forming cells. They do not involve multiple cell types, but when you put this material inside the in vivo environment, then multiple cell types will come in contact with the biomaterials, or in other words, the biomaterials will come in contact with certain tissues, and that those tissues can contain multiple cell types. Then fourth point is that there is no tissue remodeling that is possible, or that cannot, or that can be assessed in the in vitro test. Tissue remodeling means that extracellular matrix composition during in vitro tests are largely do not change with time, but inside the in vivo environment, extracellular matrix composition, ECM composition continuously changes, because in vivo environment there is more dynamic changes in pH, there is more dynamic changes in composition of the ECM as well as cell, proteins, etc. Now, those kind of things you cannot really simulate in the in vitro test. Therefore, in vivo tests are necessary, and they provide information on the following aspects, like first one, interactions of the different cell types. As I mentioned that during in vivo implantation, that a material will come in contact with different cell types. Effects of hormonal factors, those factors cannot be assessed in the in vitro conditions. Interactions with extracellular matrix, that is also important things. Interactions with blood-borne cells, proteins and molecules. Now, this, although you use some serum protein or other kind of proteins in the in vitro test, but number of proteins that are experienced or those are exposed to in biomaterial in vitro test are limited, and different biological molecules also biomaterial will come in contact, Biomaterial also will come in contact with the leukocytes or erythrocytes, because there will be always blood flow at any given locations. So all those things are not possible in the in vitro conditions, and therefore in vivo tests are necessary. So also let me remind you that what is defined as a biocompatibility, that is the ability of a material to perform with an appropriate host response to a specific applications. Appropriate host response means, like host here it is that human or animal, and so therefore appropriate host response means that in the implanted area where the material is being implanted, those areas should not show any sort of inflammation or any toxicity just because that synthetic material is implanted in that position. And in a specific application means, as I said, this biocompatibility is a broad term, and that has a different meaning for different applications, right. For blood contacting devices it is the hemocompatibility, for bone forming or bone replacement it is the cytotoxicity, tissue formation ability, all those things are important. Now therefore biomaterials must be biochemically compatible, that is the first important point, that is biochemically compatible. It should be non-toxic and non-irritable. Non-toxic means, like that cell level or gene level there should not be any toxicity. Non-irritable means it should not cause any irritation to any part of the body. Third one is that it should be non-allergenic and non-carcinogenic. Certainly it should not cause any cancerous effect, like it should not activate the cancerous cells, and if the cancerous cells are activated, that means uncontrollable multiplication of cells will take place, and which will cause the cancer to spread. Also biomechanically compatible with the surrounding tissues. Now what it means? It means that every tissue, like whether it is a hard tissue and soft tissue, you have seen earlier lectures, they have a range of properties in terms of elastic modulus, in terms of mechanical properties. Now whenever you are putting the biomaterial for a given applications, the tissues, that whether it is a bone which will surround that material, there should not be large difference between that material and the bone which is in the neighborhood of that material. And if there is a large difference in mechanical properties, particularly in terms of elastic modulus, then what will happen? There is a biological process called aseptic loosening. That means this biomaterials can be detached from the neighboring hard tissue or bone, because if the biomaterials has a larger elastic modulus, then biomaterials will carry most of the load. If the bone has a larger elastic modulus, then bone will carry most of the load, but there should not be large difference in terms of the elastic modulus. Then last point I have mentioned here, the bioadhesive contact must be established between the materials and living tissues. Bioadhesive contact means there should be some kind of biological contact. That means when a material is implanted, then if a capsule tissue is formed around the material implant, then that is what is desirable for good in vivo biocompatibility. Now these implant effects can be simulated in vivo, that is dead space can be created by the implant, and insoluble particulate materials released by implants. Now these are the factors which you can easily assess during the in vivo testing. The first one is the dead space created by implant, means like suppose you have a very long bone structure, and the long bone you cut some pieces of the bone, then you put your implant. Now dead space means if the interface is not uniform, interface means that is the space between the implant as well as the natural bone. If the interface is not continuous, then what will happen? There will be some dead space which will be available where there is neither material nor any natural tissue. Okay. So that is what is not desirable. Then in those case there will be problem with the mechanical behavior of the bone. Second thing is that insoluble particulate materials released by implants. Now insoluble particulate materials means certain particulate materials, when it is finer debris particles and so on, then they can be easily dissolved in the body fluid or body plasma. But however, if there is insoluble particulate which is released during the in vivo degradation. In vivo degradation means like inside the implant materials, the material also will undergo some kind of degradation, and under those conditions they will release some material, they will be leaching some materials, and if those materials can cause some undesirable toxic effect around the tissues. Then third factor is the interaction of biological factors with implants, and fourth one is the mechanical loading experienced by the device. Now mechanical loading can be either compression or flexural. Now, compression means when a body will be at certain part which will be loaded from two opposite directions, or flexural means it is like a bending type of situations. Now, these kind of two type of mechanical loading that are generally experienced by the device. Now, it is more kind of explained here that increased local strain due to movement of device with respect to tissue, and hyperplasia means increased scar tissue, thicker fibrous encapsulation. You remember that is the cellular adaptation process. Cellular adaptation process means there are four types of cellular adaptation process. One is atrophy, that is the decrease in cell size. One is hypertrophy, that is increase in cell size. One is hyperplasia, that is increase in cell number. One is metaplasia, that is the change in cell type, like osteoblast to osteoclast transformation, that is the called metaplasia. Now, what is mentioned here that whenever a biomaterial is implanted inside the body, then the cells which are in the neighborhood in the tissues or the bones, those cells also can see that some foreign material is implanted at that place. As a result, there will be cells also will experience some kind of different surrounding environment which is not natural. You try to understand what I am saying. So, the cells in the tissues surrounding the biomaterial will see kind of environment where a foreign material is implanted inside the body, and which is quite different in terms of composition and in terms of shape compared to the natural tissue or natural bone if it would have been there. So, therefore, there will be cellular adaptation process, like those cells which are in the neighborhood, those cells will undergo some cellular adaptation process, and one cellular adaptation process that I have mentioned is the hyperplasia, that will increase in the cell number, and if it is large number of cells will form, then what will happen? They will form kind of scar tissue, like you might have heard this word scar. So, many times some scar will appear at the patient at the implanted area, or thicker fibrous encapsulation. Thicker fibrous encapsulation means if this is a implant, so around that implant there will be fibrous tissue will be forming, and this fibrous tissue will encapsulate the implant from the surrounding natural environment or surrounding bone environment. Now, second point that has been mentioned here, that reduction in tissue strain due to implant, like if implant takes all load, then tissue undergoes atrophy, like stress shielding. What it means, as I said, that if there is a large mismatch in elastic modulus between the tissue and the implant, and if implant has much higher modulus than the tissue, then what will happen? Implant will carry most of the load, not the tissue, and as a result that the tissue will undergo atrophy. Tissue means the tissues which is surrounding the implant will undergo atrophy. Atrophy means decrease in the cell size, and if this decrease in cell size continuously take place, then there will be effect called stress shielding will take place. Now, these are the examples of the stress shielding. Now, A is that post operative image, that is the called A, and there is a B is the image of the same patient after 7 years follow up. Now, this is B. Now, what you see that this is the implant material, right, and this implant material remain the same, but what is the notice, you can see that it is now firmly adhered to this neighboring tissue or neighboring bone, but here it is somewhat get detached or loosened because of these aseptic loosening factors that I just mentioned. Now, connective tissue is one of the examples of the fibrous, and then that is the cutaneous or subcutaneous sites. Cutaneous or subcutaneous means that is you have the skin, subcutaneous means it is just under the skin, that is called subcutaneous region. These sites are chosen to assess the biocompatibility, and this type of testing is readily accessible, and thickness of fibrous capsule is a measure of biocompatibility. What is mean? And typically these tests are carried out in guinea pig. What I am trying to say here, that if you want to do some implant testing or in vivo testing in guinea pig, the best possible way is to just put the implant just under the skin, then you leave the animal for let us say 4 weeks or 6 weeks or 12 weeks, then at regular interval you take some sample out, then you try to find out that around the implant what is the thickness of the fibrous tissue that has formed. Now, if the fibrous tissue has formed progressively in larger amount, then the material is in vivo biocompatible. But if there is no fibrous tissue formed even after prolonged time of implantation, that this material is not in vivo biocompatible. But remember, same material can be in vitro biocompatible. As I said in the earlier lectures, that this is the fundamental thing that you must remember, that if a material is biocompatible in vitro, there is no guarantee that the same material will be found to be biocompatible in vivo. Okay. Now, in case of the vascular implants, like blood compatibility of materials used as a vascular prosthesis. Now, what happens that you know that if you put this implant, you can put this implant as carotid jugular vein or in some other replacement segments of the patches, and for this kind of materials, it is not that what is the thickness of the fibrous tissue that is formed, that is the indication of the in vivo biocompatibility. Instead, if the material has good blood compatibility in vivo, blood compatibility in vivo means, in the last lecture I have categorically mentioned that there should not be thrombus formation when the material will be coming in contact with the blood, and even if the thrombus is formed, then this thrombus should be carried away from the material by some continuous blood flow. So, in those two possible scenarios, you can tell the material is blood compatible. Now, this is that guinea pig experiments I just mentioned. Now, what you see that this is a typical in vivo test applied on guinea pig on extracts of the device, and guinea pig test is considered the most sensitive test. Now, what is called extracts? Extracts mean you have a biomaterial which you can ground into very finer particles, and those finer particles you can inject them just as a drug delivery thing that is injected in the subcutaneous region. Now, if there is a inflammation, like the way it has been mentioned here, that this is the area, if there is a inflammation on the skin that you can clearly take an image and see, then this material you cannot say that it is a in vivo biocompatible. Okay. Now, implantation. So, implantation means that is a sample of a predefined shape. Now, predefined shape means it can be of cylindrical cross section, it can be square cross section, it can be rectangular cross section. All these typical geometrical shape can be placed in the long bone of a mammal. So, typically all these materials, they are implanted into a mammal like rabbit, rat, mouse. Now, as I said that this is the philosophy behind choosing the particular animal model in the in vivo experiment. If a material that you develop for bone replacement or the load bearing bone replacement materials implants, then the first thing you should do the implantation test in rabbit, not in the small animal, because small animal means rat or mouse, their bones, the strength of the bones is not as strong as the way you get the intermediate animal model like in rabbit. So, this load bearing capability or the effectiveness of this material as a load bearing implant, that you cannot assess scientifically if you use the rat or mouse model. So, selection of the particular animal model is important in the in vivo test. Other thing that I have mentioned, I think couple of lectures back, that larger the animal, like rat is a small animal, mouse is similarly small animal, rabbit is little large animal, then goes to dog, sheep, and then goes to human. Now, as the size of the animal progressively increases, the complexity of the in vivo environment also continuously increases. Like complexity means the changes in pH, changes in temperature, changes in the number of proteins available, changes in different cell types, etc., etc. In terms of biological complexity also subsequently increases if you go from small animal to intermediate animal to large animal. Okay. So, if you choose the intermediate animal model, that what we call the rabbit model or rabbit experiments, then it is you can choose, you can say that this material is in vivo biocompatible in the rabbit model. Again, if a material is successfully found as a in vivo biocompatible in rabbit model, there is again no guarantee that the same material will be in vivo biocompatible in the human being. Unless until you do the test in the human, you cannot safely state that this material can be used in the human body. You understand, this is the kind of philosophy of the in vivo test. Now, after some period, the samples and surrounding tissues are examined histopathologically to determine the in vivo response of the materials. That what it means, that suppose you implant the materials at a particular area, so you take the cross section of the material along with the tissues, then you do that standard histopathological analysis just to see that whether in the surrounding the material, whether there are cells or tissues that has formed very successfully after a certain period of time. Now, how this is done, it will be more clear in the next couple of slides. Now, first thing is that you have to know that according to the ISO standards or FDA approved protocols, ISO means International Standard Organization, FDA means Federation of Drug Approval. Now, these implantation tests are the two types of implant, that is that one is the short term and one is the long term, and then short term test can be done for up to 3 weeks to 12 weeks, and then whereas long term implantation test needs to be done for up to 12 months. Now, why these two tests are necessary, these two types of tests? Now, if you want to screen a certain biomaterial whether it is in vivo biocompatible, you can do for short term period and long term period. What happens if some materials, some leaching or biodegradation takes place over the long term period, then that cannot be assessed if you do short term period like up to 3 weeks or 4 weeks or up to 12 weeks. Therefore, if you do the long term test, then you can be more sure that this material is in vivo biocompatible for the long term basis. So, implantation studies also evaluate the potential leachable substances using chronic systemic toxicity, and then blood hematology and biochemistry are also performed in many cases. So, as I said, the different animal model which can be used or which are used like mice, rats, guinea pig, rabbits. As I said, the rabbit model typically used for the kind of materials like ceramic based materials or biopolymer based ceramic composites, those kind of materials rabbit model is used. However, if the material is entirely polymer based where the mechanical properties is much inferior compared to ceramic based materials, in that case smaller animal models like rats or mice also can be used. Now, implant is first cut into size, sterilized and implanted, and then X-ray radiography needs to be carried out, and this X-ray radiography means like you have to take that X-ray image, just like you take X-ray image for that when your leg gets fractured or your hand gets fractured. Similar X-ray image you have to take at the implant site, you have to store it, then you have to see after 1 week this is the X-ray image, after 3 weeks this is the X-ray image, like that you can compare this X-ray image. Okay, this is like standard protocols what is defined as that ISO 109936, that is the implant effect. Now, test in the long bone of the rabbits. Now, this is like 1 into 10 millimeter strips of the material that can be cut and sterilized. This is placed in gauge needles, anesthetics applied to the implant site. So, it is like every test is done in a OT, like operation theater. So, you have to give anesthesia to the local anesthesia to the animal at that site, and then 4 test sample and 4 controls of the each composition needs to be implanted in the paralumbar muscle. So, what it means, like you have to use the test samples like your materials which are testing, at the same time you have to put the control samples also. Now, why control samples is necessary? Control samples means the samples where you already know that is in vivo biocompatible. You understand what I am saying? So, that with respect to the control sample, you can now assess whether a given material or the material that you have developed in your laboratory is in vivo biocompatible or not. So, that is why the control sample is necessary, and you already have seen that all the in vitro experiments also that is always positive control or negative control sample is always used. Now, test samples and controls on the opposite sides. That means that if you take one leg of the rabbit, you put your test samples. If you take another leg of the rabbit, then you put your control samples. So, these are like on the opposite legs. Then short terms, as I said, they are up to 1 to 12 weeks. Long term, you have to do this tissue response up to 78 weeks. Now, at each interval, now what would be the interval that you will be observing? That depends on whether it's a 1 week, 4 weeks, 8 weeks, 12 weeks, or it will be 1 week, 4 weeks, 12 weeks. Now, this reactive material is typically 2 to 4 millimeter thick. What is reactive material here? That is just the material and the tissue surrounding just adjacent to the material, that is called the reactive material, and no visible capsule on the control material. And histopathological examination will actually tell you that what is the inflammatory reaction to the implant and whether is a cell death around the implant. Now, if there is a cell death around the implant, then what will happen? That the material cannot be used in the real life applications. Now, this actually tells you that how you can qualitatively assess that whether a material or whether two different materials, how good or how bad they are as far as the in vivo biocompatibility is concerned. Now, you see this is your implant materials, this white contrast materials it is a different shape. What I presume that possibly this material when it was implanted, this was like a perfect geometric shape of a rectangular shape, but these edges actually indicate that irregular edges actually indicate that some part of the material has been leached away during the in vivo implantation. This is the surrounding tissues around the implants, and if you see that this is the region where this fibrous capsule has formed around the implants. Now, it is called minimal response. So, the response is not extensive, but a composition similar shaped material of different composition were also placed here, and what you can see, entire material has been dissolved in vivo. You are left with only these small parts of the material, and there is an extensive inflammation reactions around the implanted zone. So, that means this material you cannot use, this material you can accept as the roughly like in vivo biocompatible. Now, you understand that how qualitatively you can assess whether two different materials, which one you will choose for the in vivo biocompatibility. This is like dental screws which are actually put it inside the as dental restorative applications. Now, I will show you some of the experiments that we recently carried out. Now, these are the material shape typically we used in our experiments. As you can see, these are like cylindrical shaped specimen, and I know their diameter is around 2 mm diameter. So, it is extremely small size diameter, a small size, and this is aspect ratio is around 3 to 4 aspect ratio. So, 2 millimeter diameter and 8 millimeter length, that is in cylindrical cross section, those samples were used in this in vivo experiments. Now, these are the experiments which are going on in rabbit. Now, this is the long bone of the rabbit. As I said that this long bone, you have to put some holes here. These are like 3 different holes, and these holes, the moment you drill the holes, there will be also blood coming out, right, through these holes, and you have to put the material here at 3 holes in one leg. This is one leg, that is another leg which you cannot see very clearly. So, one leg you put the rabbit, another leg also you put the control specimen. Now, what you do here, that after 1 week, this is the X-ray images. Now, what you can see, this X-ray images, this is your sample, you can clearly see your samples, right, that is your hydroxyapatite, mullite or whatever samples it is. After 4 weeks, you can clearly see the samples here, and after 12 weeks, you can see the samples almost of the same size. What it means, that there is no visible in vivo degradation from the samples. If it would have been degraded in the in vivo environment, you cannot see the same 2 mm diameter cylindrical specimen after 12 weeks, okay, because then samples would have gone to half the diameter or half the length, whatever it is, okay. Now, this is one of the way to find out that in vivo degradation. I will also show you, before going into more details about the recent results, I will also go through some of the literature results where people have done several experiments on all different type of materials. Now, titanium based alloys, as I mentioned, titanium and titanium based alloys, they are highly desirable for the stem applications of this total hip joint replacement. Stem means that is the long rod. And why titanium is preferred? I said that from the point of view of the corrosion and wear, titanium is more corrosion resistant compared to stainless steel stem, for example. Now, if you see here that after 4 weeks, these materials are implanted in an animal model, and these are like 4 different composition. One is Ti6Al4V, that is titanium 6% aluminum 4% vanadium, Ti13Nb 11 zirconium, that titanium 13% niobium 11% zirconium, Ti6Al4V, that is titanium 6% aluminum 4% vanadium plus hydroxyapatite, means this is in the form of hydroxyapatite coatings. Then you have this Ti13Nb1 zirconium plus hydroxyapatite, like you have this same alloy but with hydroxyapatite coatings after 4 weeks. And what you see in this Ti13Ibm 11 zirconium plus hydroxyapatite, here it does not show much toxicity response, and you have this very fibrous tissue forms, and you can see there are different cells. These are like osteocytes and osteoblast cells. But in most of the other cases, the same size implant, because the implant size were the same in all the cases. Okay. However, this is the region that the material has been dissolved in the in vivo environment. That means all 3 materials you cannot use that well. These materials, yes, you can consider it as a in vivo biocompatible. Okay, this shows that connective tissue ingrowth into the composite ceramic at week 4. Okay, and this is the fibrous encapsulation of hydroxide at week 4, and hydroxide acid bone and bone marrow remodeling at week 8, and composite ceramic new bone formation at week 8. Let's go through one by one. Now this one actually connective tissue. Now this connective tissue, you can see that after staining, this connective tissue actually is growing inside the hydroxyapatite. This entire material is your hydroxyapatite. This entire region is your natural tissue. Now fibrous encapsulation means, as I said, that if a material is in vivo biocompatible, around the material there will be a fibrous tissue, and this fibrous tissue will be formed and they will encapsulate the material from the surrounding natural tissue or bone. The third one is that hydroxyapatite bone growth. Now this is the bone marrow and this is the cortical bone CT, and you can see that there is a nice bone formation even after 8 weeks, and this is shown as the neo bone formation, neo bone or new bone formation at the implanted region. So hydroxyapatite is well known as that in vivo biocompatible material, and this is the proof that yes, under the in vivo conditions, even after 4 weeks or 8 weeks, you can clearly see that new bone formation. So this is after 14 days or 2 weeks of the implantation on different material like Ti6L4V, PSHA coatings and DDHA coatings. Now in all these cases you can see that is the neo bone formation, and this is also the region bone formation. Now Ti6L4B, this is your implants. Okay, this is the region that is where the neo bone has been formed. This is also that another Ti6L4B, and on that there is some coatings, hydroxyapatite coatings is there, and this region neo bone formation. So what has been shown here, that hydroxyapatite, putting the hydroxyapatite coating on this titanium 6L4V also enhances this bone growth or bone formation in vivo. Okay, this is the transmission electron micrographs, and these transmission electron micrographs essentially tells you that what happens after implantation for 14 days. So here the 2 materials were we investigated. One is a plasma sprayed hydroxyapatite coating, and another one is electrochemically deposited hydroxyapatite coating. So plasma sprayed hydroxyapatite coating, this one, electrochemically deposited is this one. Now this is your coating structure, and this is your mineralized tissues. Mineralized tissues means like these tissues has lot of hydroxyapatite content. Mineral means that is again the inorganic component of the bone, that is the hydroxyapatite. And this is the mineralized tissues which are formed here, and that you can clearly see after the staining. Now there are several other metallic implants that which are used in the in vivo conditions, that is nickel titanium. This is the shape memory alloy, nickel titanium, and this nickel titanium implant, when it is put it for 13 weeks of implantation in a particular animal model, then what you see here, that large number of bone cells and collagen fibrous networks that you can see, and a little bulge cell and collagen fibers on uncoated nickel titanium implant. Now here you can see large number of osteoblast cells, and this fibrous network you can see, these fibrous networks are essentially collagen fibers. So both the collagen fibers and osteoblast cells, if they are present in the neighborhood, that will clearly indicate that you are essentially enhancing the extracellular matrix formation, as well as the bone forming cells are activated so that you can have new bone formation. However, if you compare this with this, you can clearly see that when you do not put any hydroxyapatite coating, then you don't see that well the bone cells, different bone cells, as well as this collagen network is also not that much prominent or more visible or more extensive like that in the presence of the hydroxyapatite coating. Now, how this bone tissue histopathology that is carried out in real life? Now, you have to first fix the samples in the toxicological lab, then implanted zone are cut using diamond saw. So, using the diamond saw you can cut the implanted zone. Then you have to do it for the dehydroxylation using alcohol. So, that means that serial dilution using alcohol, you do that, you get rid of that all the water molecules which are absorbed. Then you embed it in resin. So, you put it in resin and then you cut the cross section wise using the diamond saw. Then you can grind and polish the cross section samples, and subsequently you can stain and you can observe them under the microscope. Stain means it is just like etching of the metallic materials. Stain means if you do etching in the metallic materials, ceramic materials, you can see grain boundaries. Similarly, if you stain, that means you can use some particular chemical reagent, and those chemical reagent actually will stain certain cells or the extracellular matrix or the actin filaments or the nucleus or the mitochondria. Depending on what kind of stain you used, what concentration of staining you used, you can clearly see those cells and the particular features. So, typically the stains those are used in this histopathological investigation. These stains are widely used as Stevenel's blue. The Stevenel's blue, it is generally used mostly for this bone tissue staining, and then other stain used for soft tissue is that eosin and hematoxylin. The soft tissue of the soft tissues, these are like different tissues, like these are not like hard tissues or bone cells. For the bone tissue engineering, this is always Stevenel's blue which is widely used. Now, as I said just few minutes ago that that staining agents is just like etching or etchants in case of the metals. Now, different stain, they attack differently chemically distant phases or attack some specific group or site. What it means? It means that like an etchant in case of metals and ceramics, they also chemically attack the different grains and then they will reveal the grain boundaries. Similarly, here these stains attack different chemically distant phases, like mitochondria has different chemical environment compared to the nucleus per se. So, nucleus will stain differently compared to mitochondria, or nucleus will stain differently compared to the extracellular matrix or the actin filaments or the cytoskeleton, because all these phases, although they are bound in the same cell, but they have different chemical composition, and accordingly they will be attacked in a different manner by the stains. Now, what is the dehydration? How this dehydration takes place? This dehydration, first you have to use first 70% alcohol for 1 to 4 days, 80%, 96% and 100% alcohol for 2 days, and then alcohol acetone mixture, that is 1 to 1:1, that is for 1 day, and finally 100% alcohol for 1 day. So, that is the way you can dehydrate or dehydroxylate this entire tissue material interface sample. Now, what are the things normally you should look for when you look these bone tissue samples under the microscope, for in this optical microscope or fluorescence microscope or what kind of microscope or ACM? Then you want to see first is this new bone formation, whether that takes place around the implants, that is the number one. Number two, whether there is an inflammation around the implant, and number three, whether there is a bones is the osteoconductive and it has a osteoinduction property. Now, some general observation is that during initial period, let's say many times what people have reported in the literature, like let's say first after the initial first 1 week, some kind of inflammation may be observed, and that is due to the bone marrow response. But this inflammation should not sustain for 12 weeks or 78 weeks. This will go away as you give more and more time for the animal to heal from that wound. Essentially, whenever you cut a long bone, essentially you are wounding or you are giving some wounds or you are imparting some wounds to the animal, and then every wound healing process biologically will take time. Suppose you get some your heart in your legs, so there will be some blood clotting and everything. So, this blood clotting will not go just 1 hour, it will take maybe 1 day. So, similarly in the animals also this wound healing process takes place possibly a week or so. And new osteoblast cells, they appear blue in color after staining using Stevenel's blue, and you have to also see that what is the orientation of the osteoblast cells around the implant. Osteoblast cells are not like epidermidis cell lines, like they are just lined one after other. So, they are like connective tissue cells. So, they are dispersed in the extracellular matrix, but there are a lot of distances between these 2 or 3 osteoblast cells. Now also you have to see that implant material and the bone interface, that is the new bone formed, can be observed, and in some cases some matured bone or direct material bone material contact also can be observed. This is a clear indication of a good bioactive material. Bioactive material means bioactive in vivo, then this kind of observation you can make. Then matured bone cells appear brown in color. Then if you see that matured osteoblast cells, then you see the different colors, and Haversian canal appear blue in canal. These canals are surrounded by osteoblast cells. Now what is a Haversian canal? Haversian canal is like a more or less like a cylindrical channel like of things through which nutrients and bloods and foods are supplied to the cell. Marrow protein appear black in color. Now all this color sequence you have to know that how they will appear in the fluorescent microscope, then only you will be able to identify the different images. And there is a color contrast in the new bone and natural bone formation also. That is important, because how you can see that whether it's a new bone or natural bone. Natural bone means what it has existed before implantation, and new bone means what bone formation has taken place after you put the implant at the particular animal site. Now this is the entire histopathological protocols, one has to follow to get to study this kind of in vivo biocompatibility aspect of biomaterials. Now what it shows here, this is a long bone, okay, as I have described in last lecture, that is a long bone typically has a large aspect ratio, like it has a large length to diameter ratio. Now in the long bone you are cutting at different region, okay, and then you put your implants. This is the red circles, that is the implants you have put in the long bone, and then after that you take this long bone that you can cut it by the diamond cutting machine, then you put it in the dehydration, that is in alcohol. So that is like serial dilution, that you give the treatment to this bone tissue sample, serial dilution. Then you embed it in some kind of PMMA, that is polymethyl methacrylate, okay, that is a polymer. Then after you embed, then you cut the cross sections by the diamond saw, and if you cut this cross section in the diamond saw, then essentially you are seeing that both the implant and the implant bone interface. Then you take a thin section, then you polish it in that standard polishing wheel, and then you do the staining. This staining is done by Stevenel's blue because of the bone tissue engineering, and if it is for soft tissue engineering, other stains like eosin can be used. Then you can use the fluorescent microscope to observe this, what are the different features of the neighboring bone at the neighborhood of the implant. So that is the entire protocol for this bone tissue engineering or histopathological response. Now I will show you some of the results that were obtained for this hydroxyapatite mullite composites. Now these tests were carried out Suchitra Tuner Institute of Medical Science and Technology, that's in Trivandrum down south, and that test was done for short term implantation, like up to 12 weeks. Now after 1 week, this is the hydroxyapatite 20% mullite. Because why we have chosen this particular composition? Because this particular composition demonstrated better combination of mechanical properties and better combination of the in vitro cytotoxicity, bone mineralization, etc. So therefore we thought that we will do the test only on this hydroxyapatite 20% mullite, because when you are doing this in vivo experiments, then you have to also be careful, because there is an animal ethical committee or animal welfare committee. You cannot use capital N number of samples, and you cannot kill that capital N number of animals during that any in vivo study. You have to minimize the number of animals that you want to sacrifice because of your scientific study. So therefore the selection of a particular material for in vivo test also needs to be made very carefully with some scientific rationale or with some logic. So therefore we have only chosen this particular composite, and what you see here, this is your implant. Now around 50 micron, that is some tissues are forming, and this is also a different after doing this Stevenel's blue, you can see that connective tissues as well as fibroblast cells around the implant materials which is formed. And these are the examples of newborn. This is the newborn trabecule, that is the black arrow, and the newborn trabecule means that is the trabecular means spongy bone, and that spongy bone structure you can see here, that is like a more like a porous structure. So as you know the trabecular bone, spongy bone or cancellous bone, they are synonymous words, right? They carry the same meaning. Now here you can see this is the more like cancellous bone that forms. So, and this is the neo bone that is formed in that, and this is the cortical bone, and this is the implant site. This is after 4 weeks. What you can see here, this is your material, this is your new bone. There are some regions where the Haversian canals are there, and you have this cortical bone, that is your natural bone. So you have this natural bone. This is the entire interface, that is the bone material interface, and this is your material. So that means this material shows good in vivo biocompatibility. And you can see here that around this implant zone here and this part, you can see that clearly the tissue formation, and there are also multiple different cell types which stain differently in the when you use this Stevenson blue. You are seeing some blue colored cells, also you can see some differently colored cells here. This shows that how this bone material interface they look like in the scanning electron micrograph. If you closely observe, then you can see there are some canals here, right? And these canals are essentially what I call is that Haversian canals. And also this interface is continuous. You do not see any gap in the interface, or there is any loose part in the interfacial region. This is again some new bone formation, and this cortical bone is formed, and this is your implant material. Often if your sample preparation is not good, then this is a brittle material you are handling, you can cause some crack at the interface. These cracks are not necessarily formed because of any in vivo reaction. So you have to also analyze it carefully, because you are handling the ceramic based material which is brittle, you are handling some bone tissue at the interface that is also brittle. So if your sample preparation is not done with extreme care, often you do see some kind of cracks at the interface. This is after 12 weeks, and 12 weeks also you can see this is the shape of the implant, and then from this shape of the implant you can notice that this material does not undergo any in vivo biodegradation process, and these are like different cells, like these are like osteocytes, osteoblast cells. Now osteocytes is the white arrow, and new bone is the black arrow in different images, that is fluorescent images that you see here. This is 12 weeks. This is very good picture. What you can see, this is a material, this is your new bone, and that appears in different contrast, and this is your actually natural bone. Natural bone, again natural bone contains both trabecular bone as well as the cortical bone. Typically the surface, outer surface is cortical bone, inner surface, inner area is the trabecular bone or the spongy bone structure. So I think I have finished here. That is the discussion on the in vivo implantation, and then in the next lecture I'll start with some of the results that we have got, experimental results that we have got on this hydroxyapatite based different biocomposite and some of the polymer ceramic composites.